Scientific inquiry, methodology, techniques, and history. First, we'll go through a summary of scientific inquiry, methodology, and history. And then we will work on some practice questions related to that you might see on the biology praxis exam. Scientific inquiry, processes, and social perspectives summary. Scientific inquiry processes. There are many different processes, but here is a representative one. Make an observation that is objective and may be duplicated by other scientists. Gather and evaluate available information about the observation. Make an educated guess or hypothesis to explain the cause of the observation. It must be testable. Conduct an experiment to test the hypothesis. Analyze the data and report if the hypothesis was supported. Share that data with others. Make a new prediction or make new predictions based on the data analysis. The words theory and law have specific meanings in science. A theory explains why many types of physical phenomena occur and has been repeatedly tested by many researchers and found to be valid. A law describes physical phenomena and cause and effect relationships. It makes no claim as to why the phenomena behave as they do. Like a theory, it has been repeatedly tested and found to be valid. Experimental design. Experiments need to be carried out without bias. No matter how elegant a hypothesis may be, if the experiment shows it is false, then a new one is required. Once the problem is identified and a hypothesis is created, the experiment can be designed using prior research as a guide. The experiment should vary only one variable at a time to ensure that the result depends on that variable. All the other quantities should remain the same, the control variables. The independent variable is the one that is altered. The dependent variable occurs based on the independent one. Analyze the data without bias towards your expected result. Share the data with other experimenters. If they cannot reproduce the experiment and the results, then it cannot be used to support hypotheses until this is corrected. Nature of scientific knowledge. Scientific knowledge is based on questions that can be answered by experiment, detailed observations, or calculations. The knowledge should lead to models, theories, and laws that are used to make predictions about related phenomena not yet observed. If the predictions fail, then the models must be modified to fit the new results. The best models unify different, previously separate observations, like electromagnetic theory combined electricity and magnetism principles. Scientific knowledge is always subject to change as more complete experiments or observations are made. As Einstein's special theory of relativity modified Newton's 3 laws for objects moving near the speed of light. Scientific models. In addition to explaining phenomena with theories and laws, scientific models are also used to make natural phenomena more understandable. For example, the structure of cellular membranes is described as a fluid mosaic model, since the many structures in the membrane can be thought of as different shaped and colored tiles in a mosaic, and they move around as if they were in a fluid. Electric current is modeled as a flow of water, since electrons are flowing through wires. A limitation of this model is that water flow does not need an unbroken or complete path to flow where electrons need both. Biology principles and historical figures. The source for this topic is this website here, www.famousscientists.org, and we're looking at top biologists. Below, it's organized roughly by date, which also correlates with the principles which each scientist helped launch or explore. In the 16th century, Andreas Vesalius founded modern anatomy, overthrowing misconceptions about the body that had persisted for over 1,000 years. In the 17th century, Robert Hooke discovered cells and wrote one of the most significant books in scientific history, Micrographia, revealing the microscopic world for the first time. Antoine van Leeuwenhoek, the father of microbiology, used remarkable self-made lenses to discover single-celled animals and plants, bacteria, and spermatozoa. Also in the 17th century, William Harvey explained blood circulation for the first time, showing there's a complete circuit beginning and ending in the heart. In the 18th century, Carolus Linnaeus organized our view of the natural world with the two-part naming system we use to classify all life forms. Named and classified about 13,000 life forms, broke with tradition by classifying humans in the same way as other life forms. In the 19th century, Theodor Schwann established that the cell is the basic unit of all living things. His classification of cells is the foundation of modern histology. He discovered the enzyme pepsin and identified the role microorganisms play in alcohol fermentation. Continuing in the 19th century, Gregor Mendel founded the science of genetics, identified many of the rules of heredity, identified recessive and dominant traits, and that traits are passed from parents to offspring in a mathematically predictable way. Charles Darwin authored one of the most famous books in history, On the Origin of Species, in which he described and provided evidence for the theory of evolution by natural selection. Alfred R. Wallace independently formulated the theory of evolution by natural selection and was one of the first biologists to express concern about the effects human activities were having on the natural world. Louis Pasteur, the father of modern microbiology, transformed chemistry and biology with his discovery of mirror image molecules. He discovered anaerobic bacteria and established the germ theory of disease, invented food preservation by pasteurization. In the 20th century, Maurice Wilkins initiated the experimental research into DNA that culminated in Watson and Crick's discovery of its structure in 1953. Maurice Wilkins crystallized DNA and obtained the best quality X-ray images of DNA seen at the time, indicating DNA molecules were helical. Rosalind Franklin provided much of the experimental data used to establish the structure of DNA and discovered that DNA can exist in two forms. Francis Crick and James Watson are the co-discoverers of DNA's structure and replication mechanism. They established the sequence hypothesis and the central dogma, and discovered that DNA uses a triplet code to control the formation of proteins from amino acids. Ronald Fisher invented experimental design, devised the statistical concept of variance, unified evolution by natural selection with Mendel's rules of inheritance, so defining the new field of population genetics. Continuing in the 20th century, Stephen Jay Gould devised the theory of punctuated equilibrium, which proposes that evolution consists of long periods of stability broken by shorter periods of rapid change. He's an award-winning author and popularizer of science. Barbara McClintock did groundbreaking work in genetics. She showed that genes switch the physical traits of an organism on or off, discovered chromosomal crossover, which increases genetic variation in species, and discovered transposition, that genes can move about within chromosomes. Linus Pauling, a maverick giant of chemistry, formulated valence bond theory and electronegativity, founded the fields of quantum chemistry, molecular biology, and molecular genetics, and discovered the alpha helix structure of proteins, and proved that sickle cell anemia is a molecular disease. Alfred Hershey and Martha Chase carried out a series of experiments that confirmed DNA was the genetic material using radioactive labeling, bacteria, and bacteriophages. Carl Woese discovered a third basic form of life, the archaea, redrew the tree of life, revolutionized biology using genetic analysis, allowing all forms of life to be included in the study of evolution. Rachel Carson, a founder of 20th century environmentalism, her book Silent Spring led to a reappraisal of the effect of chemicals such as DDT on the environment, leading to bans and heavy restrictions. Science and technology impact on society and the environment. Science and technology have brought many benefits and many risks to society and the environment. Sometimes both arise from the same technology. Decisions on the trade-offs between the benefits and risks have to be constantly made. Nanotechnology has applications that include repairing damaged human tissue, improving the efficiency of solar energy production, and delivering drugs to certain cells. Chemistry will show how two drugs interact with one another inside the human body, playing a vital role in pharmacology and the synthesis of different pharmaceuticals. Acid rain happens due to a chemical reaction that occurs when compounds such as sulfur dioxide and nitrogen oxides are released into the air. In medical imaging, nuclear magnetic resonance happens when the nuclei of atoms within the body begin vibrating when under the influence of radio waves, which is a technique used in the medical field to detect tumors in soft tissue. Air pollution occurs when particulates, harmful gases, or biological molecules are released into the atmosphere. Water pollution happens when foreign objects and chemicals enter the aquatic ecosystem. The greenhouse effect is a process where radiation from Earth's atmosphere warms its surface to a temperature above what it normally would be. Halogen radicals that are released into the atmosphere act as a catalyst in the breakdown of ozone, causing ozone layer depletion. Chemical recycling involves chemically reducing a polymer to its original monomer so it can be processed and made into new plastic materials. Energy use and production. The generation of usable energy in great quantities for society was a major achievement of the 19th and 20th centuries, but it has issues. Energy resources are divided into renewable, hydropower, geothermal, biomass, wind and solar, and non-renewable, oil, gas, coal, and uranium sources. All have an impact on the environment in different amounts. Hydropower is very clean, but in some cases, communities are moved to make way for a dam. Wind and solar are mostly clean, but both are not consistently available. Burning coal, oil, and gas cause air pollution and increased levels of CO2 in the atmosphere, but are reliable and efficient. The transportation of fuels can be risky, and the waste from each process needs to be managed. Data collection, analysis, and sources of error. Data organization and presentation. Data should be collected in an organized fashion and should never be removed if it is not believed to be correct. Later analysis might show that an unexpected event has occurred. If the data are found to be incorrect, they should not be presented, but they must be saved. Calculations should be complete and accurate. Raw and analyzed data should be presented in tables, charts, or graphically so that the readers can clearly see what occurred. Experimental results must be analyzed quickly, yet very carefully so other groups can examine and reproduce the data. The 7 base units contained in the International System of Units, SI, should be used. Meters for length, kilograms for mass, seconds for time, amperes for current, Kelvin for temperature, mole for amount of substance, and candela for light intensity. Exceptions are for specific research where more experimentally relevant units are used. For example, in optics, nanometers, or 1 × 10⁻⁹ meters, are used to measure wavelength. Derived units are a combination of the base units, such as velocity is meters per second, and force is kilograms meters per second squared, or a Newton. Dimensional analysis converts less familiar units into more workable ones, typically SI units. process uses a sequence of unit conversion factors, each of which is equal to 1. In this case, we're going to convert furlong per fortnight to meters per second using a series of unit conversion factors. The first being 2.012 times 10 to the second meters per furlong. And then 1 fortnight in every 14 days. And then 1 day has 24 hours, and then 1 hour equal to 3,600 seconds. And in doing so, we can see that the units that we're left with after this dimensional analysis is meters per second. Scientific notation and unit conversion. Scientific notation expresses numbers as a base, normally an integer, followed by a decimal and more integers to the right of the decimal, times a power of 10. So you can see the number 336,020 expressed in scientific notation, 3.36020 times 10 to the 5th. Scientific notation uniquely specifies how many significant figures, which we'll explain next, are in the measurement. All the numbers shown in scientific notation are significant. Unit conversion is the process of using a ratio equal to 1 to convert a measurement to a different unit. Here you can see the conversion from 1,000 centimeters to meters, knowing that 1 meter is 100 centimeters. Significant figures include definite values and one approximate value, which is the last digit. Non-zero digits and zeros located between non-zero digits are always significant. Zeros to the left of all non-zero digits are not significant. Zeros to the right of all non-zero digits are significant if there is a decimal point either before or after the zeros. Multiplying and dividing numbers will have the same number of significant figures as the number with the least number of significant figures. The result of adding and subtracting numbers will have significant figures up to the rightmost column where all the numbers have a significant figure. Propagation of uncertainty or error. When data are collected, there's an uncertainty associated with each measurement due to the device. The uncertainty gets greater or propagates when the data are used in calculations. So shown here on the screen are the basic equations that we need for the calculation of error for the addition of variables. So if our end result here is given as R is dependent on these two variables, X and Y, right? A certain experiment requires multiple instruments to carry out, and those instruments each have different variability in their measurements. The results of each instrument give us these results X and Y. And the end result is R. The calculation of this error is a differential calculus problem, okay? But it results in this exact formula for the propagation of error and what we see here as the standard deviation. And this is often what's reported is the standard deviation. Again, there are some more complex calculations, but they're beyond the scope of what we need for this praxis exam. Sources of error. Measurements are not perfect. Sources of error give rise to uncertainties in the measurement. Procedural errors are not valid sources of error. The experiment should be repeated with the correct process. Systematic errors are due to improper use of a measuring tool or improperly calibrated or damaged instruments. The accuracy of the measurement, how close the measurement comes to a generally accepted value, is affected by systematic errors. Random errors are caused by changes in the measuring instruments or the environment. The precision, how close multiple measurements of the same quantity are to each other is affected by random errors. Precision and accuracy. When discussing measurement errors, these two terms are sometimes incorrectly used to describe the same concept. This example indicates their correct use. Accuracy. An experiment determines that the speed of light is 3.01 times 10 to the 8th meters per second. The value that is accepted by the scientific community, as it has been verified countless times by numerous researchers, is 2.99792458 times 10 to the 8th meters per second. The experimental value is then considered very accurate. Precision. If the light experiment measured 5 values for the speed of light, from 3.56 to 3.55 to 3.54 to 3.52 and 3.53 times 10 to the 8th meters per second, they're considered to be precise, but not accurate. Valid experiments should be both accurate and precise. Data analysis. Data analysis is a set of processes for gathering, inspecting, organizing, and displaying data with the goal of discovering useful information that will either add support or diminish support for a hypothesis. The requirements for proper data collection must be clear and unambiguous. Understanding where data comes from and how it is collected is of paramount importance. Data represented in tables and graphs. Once the data are collected and presented in tables, several mathematical tools can be used to understand trends and relationships in the data. The data can then be plotted with different graphs that are selected based on the experiment. Measures of central tendency. The mean of a data set is the sum of numerical data values divided by the number of data values. It is often called the average. The equation is given on the AP Biology reference sheet as follows, where the mean value is the sum of all the values divided by the total number of values. The median of a data set is the middle data value when the values are written in numerical order, typically from smallest to largest. The mode of a data set is the data value or range of data values that occurs most often. Mean, median, and mode. Data were collected for the age of the Apollo mission pilots and placed in a table. The mean, median, and mode are found as shown, of the ages for the Apollo pilots given below. So, the mean is 38 years. If we add them all up and divide by the total number, 7. The median is 37 years. That's the middle value. And there are 2 modes because there are 2 values for 36 and 2 values for 37. These are the 6 types of graphs which you will need to be able to create or interpret. You need to be able to determine the best graph to use based on the type of data. Scatter plots and line graphs are used when the value of each variable depends on the value of the other, and each variable can be considered as continuous over a range of values. Bar graphs and pie graphs are used to represent discrete data. The variables are not continuous, nor do they necessarily span a wide range of values. Histograms and box and whisker plots show the distribution of continuous values of a single variable for data which may or may not be normal, i.e. parametric. Parametric data will be discussed later. Scatter plots and line graphs. This set of data shows that for each value of one variable, there is a corresponding value of the other. For every math grade, there is also a science grade. It implies that the variables are defined over a wide continuous range. Whether there is a pattern relating the variables to one another is best determined by first graphing the points. The line provides the relationship between the variables and implies that it is the same between and beyond the measured values. So you can see here the science grade plotted on the y-axis and the math grade plotted on the x-axis by these green dots. Whether that is true would need to be confirmed by further experiments. Once the line is found, the relationship between the variables is fully described by the line's slope and intercepts. Now let's do an analysis of an alternative set of data. Plotting the points for this set of data leads to a different conclusion about the best graph. So we have another set of data for math grades and science grades. Let's take a look at it. You can see the points shown here. These points reveal more detail than can be captured by a single line of best fit. Instead, a line graph will be used. In a line graph, all the points are connected by lines. This graph implies the relationship between the values between points on the line connecting them, as you see in blue, but not beyond the range of the points. Conclusions can be drawn from this graph, but the relationship between the variables of science grade and math grade is more complex than could be captured by a line of best fit. Pie graphs and bar graphs. Both bar graphs and pie graphs can be used to present discrete data. Here, the same data is presented with either type of graph. This is U.S. energy consumption per sector in 2007. Presented as a pie graph and as a bar graph. The bar graph is a great way to represent data from experiments in which the independent variable is discrete, not continuous. No relationship is implied by the order of placement of the substances along the x-axis. This means that the independent variable is discrete and not continuous. So we're talking about the independent variable of methyl alcohol for this bar, water for this bar, ethyl alcohol for this bar, and acetone for this bar, but not implying any sort of continuous relationship between these substances. Compare and contrast bar graphs and histograms. Data in different categories can be displayed using bars with lengths that represent frequency. Histograms show data in intervals along a continuum. So you can see here the continuum of age from 20 to 100, whereas bar graphs show a specific value for a specific category and have space between the bars to show that those categories are separate. Box and whisker plot. A box and whisker plot is a data display that organizes data into 4 groups. Each group represents 25% of the data, known as a quartile. Quartiles are formed by calculating the median, lower quartile, upper quartile, and the extremes, both the lower extreme, which is the least data value, and the upper extreme, which is the greatest data value. The entire box here represents 50% of the data. 25% of the data lie in the box on each side of the median. Each whisker represents 25% of the data. Normal parametric distribution. Many different aspects of life fit this type of distribution. Imagine graphs for the height for humans, weight of bears, or size of homes. Most data would be around the same number, the mean, yet there would be some that would be larger or smaller. Finally, you would have the extremes that would be rare. This is called a normal distribution. Normal distributions are also called parametric because they're completely defined by 2 parameters, the mean and the standard deviation. The histogram of normally distributed data is unimodal, with the mean, median, and mode sharing the same central value. A box and whisker graph of this data is symmetrical, with the median falling exactly midway between the upper and lower quartile and between the lower and upper extremes. Much of the variance of data found in experimental results is parametric. This shows the number of distribution of data in a set of parametric data as a function of the distance of each value from the mean. The horizontal axis is measured in integral numbers of standard deviations from the mean. We will explain that standard deviation in coming slides, but if this is the mean, then this is values within one standard deviation of the mean in either direction. Over here, encompassed by this area, are all values within 2 standard deviations of the mean. And out here at the extremes, we would encompass all of the values within 3 standard deviations of the mean. In normal distributions, the area under the curve is used to calculate percentages or probabilities. These percentages are the same for all normal distributions. About 68% of data falls within 1 standard deviation of the mean. 95% of data falls within 2 standard deviations of the mean, and nearly all, 99.7% of the data, falls within 3 standard deviations of the mean. Indicated below are the percentage of data which fall within each portion of the parametric curve. In this case, the figures are rounded. So you can see that 34% of the data lies within 1 standard deviation of the mean. Between 1 and 2 standard deviations of the mean is 14%, so that all of the data here encompasses, this is 48 on one side and 48 on the other. So 96% of the data within 2 standard deviations of the mean, plus an additional 2% here, is 100% of the data within 3 standard deviations of the mean, if we round the figures. Standard deviation. Standard deviation is calculating using the following formula, which is provided on the AP Biology reference sheet as follows, where the standard deviation is the square root of the sum of all of the values minus the standard deviation squared over the number of values minus 1. To calculate the standard deviation, find the mean by adding up all the data points and dividing by the number of data points. Find each data point's deviation from the mean. Subtract the mean from each data point. Square each deviation from the mean that you find in step 2. Then add all those squares and calculate the variance, or divide that sum by n minus 1. That's the variance. And then calculate the square root of the variance. Experimental data is often parametric. The accuracy of the measurement is higher if the standard deviation is lower, since there is less variance between the measurements. They're all closer to the mean. Calculating the standard deviation of a data set provides a key measure of how accurately the data set determines the mean. When comparing the means of 2 data sets, a key element is whether differences are due to the distribution of the data within each data set or are due to a real difference in the means of the 2 data sets. Standard error of the mean. The standard error of the mean is critical in determining whether measured differences are statistically significant or are due to random variance in the measured data. It is calculated from the sample standard deviation S and the sample size N using the formula provided on the AP Biology reference sheet, where the standard error of the mean is equal to the standard deviation divided by the square root of the number of values in the sample. The standard error is always lower than the standard deviation, assuming more than one measurement is made, and is inversely proportional to the square root of the number of measurements made. Increasing the number of measurements will reduce the standard error. For instance, making 4 times as many measurements will halve the standard error. Recall that for parametric data, 68% of the data falls within 1 standard deviation of the mean, and 95% of the data falls within 2 standard deviations of the mean. Those same figures hold for the standard error, whereby 68% of the data falls within 1 standard error, and 95% of the data falls within 2 standard errors. Increasing the sample size n decreases the standard error and more precisely locates the mean. This is seen graphically in error bars. Error bars are drawn one standard error above and one standard error below the value of the mean if knowing that there is a 68% probability of the actual value of the mean being within that range is sufficient. Error bars are drawn two standard errors above and two standard errors below the value of the mean if knowing that there is a 95% probability of the actual value of the mean being within that range is sufficient. Each bar in the graph represents the set of values measured for the dependent variable in response to a different value of the independent variable. The values of the dependent variable have a mean, represented by the blue bar, and a standard error of the mean, SE error bar. The top of each bar in blue is the mean value of the dependent variable for a given value of the independent variable. The height of the error bar is 4 standard errors. It extends 2 standard errors above the mean and 2 standard errors below the mean. If the error bars overlap for each value of the independent value, then there's no statistically significant effect due to the change in the independent variable. That is the case with these data. The top of each bar is the mean value of the dependent variable for a given value of the independent variable. And you can see the overlap in the bars shown. In this case, there's not a statistically significant difference between the first 3 bars. But there is a statistically significant difference between the first and last bar, since their error bars do not overlap. There's a 95% probability that the values for the first and last bars are different.